So just a little running order for today's webinar. I'm just going to start with a brief introduction to some of the sort of key concepts behind the work that Daniel will be talking about for the majority of today's webinar. He'll take you through the motivation for the work he's been doing, looking at a comparison of correlation and importance analyses, and draw a few conclusions before we wrap up at the end with some Q&A. So first of all, just a few words of introduction. And the sort of overarching theme of what we're talking about today is addressing some of the key challenges that we have with the data that we deal with in drug discovery. Many of these will be very familiar to you. We're in a project trying to go from our initial concept to candidate faster and progress the compounds through that process as quickly and as efficiently as possible. One of the challenges there is that we can't measure all of the compounds of interest in every experiment of interest. It's just too time-consuming and expensive. This is the challenge in data science terms of data sparsity. Most of the data we would like to have hasn't actually been measured in practice. So how do we actually make the most of the data that we do have? Of course, during the optimization of compounds during a project, we're trying to identify better compounds. One way that we can be led astray is the variability that we have in the experimental data that we have measured. We know there are false negatives that can lead us to miss valuable opportunities, and false positives that can lead us to waste a huge amount of time and effort, only to discover that the hypothesis we were exploring was based on faulty information. And we'd like to explore as many optimization strategies as possible in the search for the sort of sweet spot in chemical space where we're going to find novel compounds with the best chance of success for my project. And along the way, we want to make informed decisions. We'd like to understand what's driving the efficacy, disposition, and safety of our compounds. But the data that we have are very complex and, as we've said, very noisy, and this can mask those important relationships that can help us to understand those factors. And also, we'd like to inform the decisions we're making in terms of the experiments to do. Given that we can't do every experiment on every compound, what new experimental data, if we generate it, will give us the most information with which to make good decisions about progression of compounds? Now, many of you will have joined us for previous webinars, and we've covered many of these subjects in past examples, past case studies and webinars that we've given. So today, what we're actually going to be doing is to focus on the final 2 points here, looking at how we can actually use the data we have to better understand those relationships and hence make better informed decisions. Now, before we get into that, I just wanted to talk a little bit about the methods that we have, that we're using, the method particularly for deep learning for imputation of data, and contrast imputation with more conventional QSAR or quantitative structure activity relationship models. So when we're building QSAR models, we start with a compound structure, and from that we can calculate a set of descriptors based purely upon that structure. Now, of course, we don't do this just for a single compound, but in practice, we actually calculate descriptors for a whole set of compounds. And then we construct a model that builds a mathematical relationship between those structural descriptors and the experimental data that we have for those compounds for a property of interest. Now, of course, with multi-target models, we can go further than that and build a model that captures not just a single experimental endpoint, but actually makes predictions for multiple experimental endpoints simultaneously. And if we want to make a prediction for a new compound, we can take that structure, calculate the same descriptors for that compound, and use that as input to our model to make predictions for that new molecule across the properties in which we're interested. Now, imputation is quite a different process. This is about learning from the relationships between different experimental endpoints to fill in the missing data. So here what we have are the descriptors, which we can calculate, so we can still capture that structure-activity relationship. But we also have a sparse matrix of experimental data. We have multiple experiments we're looking at, multiple endpoints that we've measured. And the green squares here represent the measurements that have actually been made. But as I just described, most of that matrix is empty. It's white space because we can't measure all of the data for all of the compounds. But an imputation model learns about not only the relationships between the structural descriptors and those different experimental endpoints, but between the experimental endpoints themselves to understand those key relationships. And so if we then want to make a prediction for a new compound where we have a small amount of experimental data, so again, we can calculate the descriptors. We have, in this case, just two experimental data points that are shown by the green squares. We can use that imputation model to fill in or impute the missing data on the right-hand side here. Again, those purple squares. And of course, we can do this for more than just one compound. We can actually take an entire matrix, entire data set of compounds with sparse data and fill in all of that data that's missing using an imputation model. The particular method that we use in practice is called Alchemite. This is developed in partnership with our colleagues at Intelligence, another Cambridge-based company. And as I mentioned, it learns directly from the relationships between experimental endpoints as well as structure-activity relationships. And hence it makes better use of that sparse and noisy experimental data that we have in drug discovery than conventional QSAR models. It fills in the gaps in the data much more accurately than QSAR predictions. We've demonstrated that in numerous publications and presentations, some of which you can see at the bottom of the slide, and more are at the end of the presentation as well, so you can look at those in detail. But ultimately, this enables us to identify high-quality compounds with the profile of properties that we need in a safe and efficacious drug for our project. But why is this particularly relevant to drug discovery data? And that's because the data we have in drug discovery is sort of illustrated, the pattern of data is illustrated here. What we have is a matrix, as I showed you before. Across the top are different experiments. Down the side are the compounds for our project. And as we go from left to right across the top here, we're going from early stage, simple in vitro, biochemical activities, simple physical chemical properties, and so on. These are higher throughput experiments and cheaper to run, and they represent simpler properties of these molecules. But the information is less informative to find a high quality sort of candidate compound for our project. It's further away from that ultimate therapeutic indication. On the right-hand side are those late-stage, potentially even in vivo experiments. These, of course, are much lower throughput, much more expensive to run, and represent much more complex properties. But of course, these are the most informative. They translate best into the ultimate clinical efficacy of a compound. And as we go from left to right, we have data increasing sparsity. We typically have most of the data for the compounds on the left hand, with experiments on the left-hand side, but as we move from left to right, of course, we have less and less data for those compounds. Now, those are the most informative endpoints on the right-hand side, but they are also almost impossible to reliably predict with QSAR models. Why is that? Well, those are the most complex properties that we're interested in. Many different factors govern the ultimate outcome in those experiments. For example, it might be one or more target activities, physicochemical, ADME, PK properties, all need to come together in terms of an efficacy. So it's very complex to predict. So if we wanted to do that based purely on structure, we would need an enormous amount of data to dissect those different factors governing that ultimate outcome. But of course, the catch-22 here is that is where we have the least data because it is the most low throughput and expensive experiments to run. So this is where imputation can really help because that early stage data contains very, very valuable information with which to impute those downstream complex endpoints. In other words, we can sort of sweep across this matrix, again, the purple squares, and fill in those blanks, leveraging not only structure-activity relationship, but those early experiments that we've done to accurately predict those downstream, expensive, but very informative outcomes. The other advantage is that as we are building this model, it is capturing very valuable insights from the relationship between those endpoints, even though that data is sparse. And again, today, what we're going to be focusing on is how we can actually leverage those relationships to gain those insights and use those to inform the decisions that we're making. One way we can do that is to actually understand those relationships between the different endpoints. So this Cerella identifies these nonlinear correlations between these endpoints from the sparse data. And there's an example here on the right-hand side where we're looking at the importance of different early stage endpoints to predict the in vivo efficacy of a compound in an animal study. And so the height of the bar, the intensity of the color relates to the strength of that relationship, that correlation that it's identified. So you can immediately see here this very tall red bar is identifying one particular target activity that is driving that in vivo efficacy. Though there are possibly a couple other targets involved as well. Certainly on the left-hand side, you can see a physical chemical property that's very important. And there's some PK data from two different species. And you can see here that species 1 has a greater importance than species 2. So it is more related to that ultimate in vivo outcome. So we can look at this for an individual endpoint like this in vivo efficacy. We can also sort of roll this up, if you like, into a single sort of row here where just the intensity now is showing the importance of each input to that in vivo efficacy. And we can generalize that across all of the modeled endpoints that we're using as inputs here. And this helps us to really understand all of those relationships driving the different outcomes that we're predicting. And this is what we call the importance matrix, and is something that Daniel will be talking about more as we go through the presentation. And once we understand that importance, we can also use it to drive decisions around the experimental data to measure next, to make more informed decisions. So it uses those relationships I just described, along with an understanding of where the uncertainty is greatest in the data and in the predictions it's making, to say, here are the most valuable measurements to make, which then, if used as input to the model, will allow us to make even better predictions of those downstream outcomes. This helps us to more confidently progress only the best compounds downstream to those expensive experiments, and hence maximize the return on investment in the experimental resources for your project. So hopefully that helps to set the scene regarding the study that you're about to hear from Daniel and some of the capabilities of our Cerella platform. And now it's over to you, Daniel.
Thanks, Matt. Hey, everyone. I'm happy to be here with you. I'd like to start just by talking a little bit about the motivation for the study that we'll examine today. So one of the things that we've seen, I think, in the past couple of years, in fact, many of them have even made their way into the popular literature about how machine learning methods can often be obscure and black box, occasionally even produce sort of ridiculous results, right? And this gets lots of hype in the media, but it arises from the fact that not all of our machine learning or artificial intelligence methods are explainable. Right? Sometimes we've got methods that use so many thousands or millions of parameters that it can be difficult to ascertain on what basis it's making a particular prediction or giving a particular result. So there's been growing interest in what we call explainable artificial intelligence. And here the focus is not just to have a good model, but to also understand how the model is working. What is it using to build the predictions or results? Uh, of course, this helps us to have confidence in applying the results, right? One of the big benefits of explainability is that we can understand what the model is doing in a way that gives confidence in the results. And as Matt has just been speaking to us about, these explainable approaches will help us to identify the important relationships that are hidden in our complex data. This is where these methods really excel, and we'll look at some examples of that through today's case study. It's important to note there's often a trade-off between explainability and accuracy. Some of the methods that focus most on explainability will use only a handful or maybe a dozen parameters, and that can limit the power of the model. Whereas we've mentioned some of the larger models with millions of parameters can give a wider variety of results, but at the cost of interpretability. What is it that's giving rise to those particular outputs? So as Matt's been talking to us about, right, we can think about imputation, this process of deep learning imputation, and how it helps us to predict results better and to be more confident in the results that we predict. There's a beautiful study by Whitehead cited here where they looked at the coefficient of determination. So that would be the R squared fit to the identity line. And the coefficient of determination can be improved as much as 0.2 for imputation over QSAR methods. And what we think this means, right, the better predictions, the better results, the more accurate models are arising from capturing those more important relationships in the data, as Matt was just talking about, and as we'll look at in detail in the next few slides. So the motivation then for this study is to say, well, okay, this is great. We've got these machine learning methods. Are they really providing value beyond what we would see from a standard correlation analysis? So the method that we'll explore in this little case study is that we'll compare correlation analysis and importance analysis for our imputation model on the same data set. And that data set is the pathogen box data for medicines for malaria ventures. It includes about 400 compounds and 50 endpoints covering a range of ADME, toxicity, and activity for anti-malarial and anti-tuberculosis outcomes. So a small data set, but enough to have some interesting relationships. And so we want to see what's the difference between a standard correlation analysis and the importance matrix that we get from this deep learning imputation that Matt has described for us in the background. So just a few notes. Technically, it's straightforward to calculate the Pearson correlation coefficient for all endpoints in the data set. In order to ensure comparability between the correlation output and the importance matrix that we get from our deep learning, we need to account for endpoints that we call they're grouped. Anytime the endpoints are measured together, we don't want our deep learning method to try to impute two endpoints that are run as part of the same experiment. So to ensure that our results are comparable, we've manually zeroed out those correlations for the assays that have been identified as being in the same endpoint group in our deep learning imputation. So then, as Matt described, we want to compare that sort of standard correlation analysis with the importance matrix that we get from our deep learning imputation attained from our Cerella platform, which we'll explain a little bit more about that and how you can experience that towards the end of the webinar today. So again, we've seen this matrix before. We have inputs here now along the vertical axis and outputs along the horizontal axis. If we assume that these are ordered chronologically, the way Matt described in the introduction, then this point that's circled here, this dark red point towards the top right of this matrix, represents a point where an early stage input is very important for predicting a late stage output. And this is exactly what Matt was describing. This is the real interest, the real power of these deep learning imputation models to be able to help us use data that's easy to collect to confidently predict results for endpoints that are much more expensive and much more difficult to measure experimentally. So to compare these two methods, we'll look at the top 10 most accurately predicted endpoints. So of the 50 or so endpoints in this data, we can rank order them according to the imputation model's accuracy against a held out test set. And so we'll look here at the endpoints that are most accurately imputed by our deep learning method in Cerella. And we'll see how that compares to the information that we'd get from a standard correlation analysis and what value is added by our deep learning methods. We'll just say that over these 10 endpoints, the average R squared, again, that's the coefficient of determination. So the identity line now is what we're considering. The average R squared across these 10 endpoints is greater than 0.7. And if we look at the top 5 most significant endpoints from each method, only about half of them are in common to both methods. So of the relationships that the correlation approach and the deep learning imputation approach identify, only about half of the relationships are the same in identified by each method. We'll continue to focus on those 10 most accurately imputed measurements as we work our way through the case study. Before we dig in too deep on the results, I want to take just a moment and follow on with what Matt was talking about earlier and how we can look at the importance matrix. And I'd like to present just a schematic representation of the data we'll show and why we think this is a powerful visualization. So we have two methods that we're trying to compare. You can see here on the horizontal axis, we're showing the results of our correlation analysis. So we can calculate the normalized absolute value of the correlation coefficient, and that will be plotted on the horizontal axis. On the vertical axis, we'll plot the importance values from the importance matrix that's derived by Cerella from the deep learning imputation model. So the idea here is that if the importance and the correlation give the same value, we'll see those along that identity line diagonally across the middle of the graph. And we'll be able to see then if there are particular relationships that one method is identifying as more significant than is captured by the other method. So with that introduction, then let's turn into the details of the case study and examine some of our results. As we look at the results from our analysis, we find that the results break down into what we might consider 4 classes. Some of these classes were described by C and co-workers. This is a paper for Medicines for Malaria Ventures. They did a big study. Optibrium was privileged to be part of that study and contributed the most active prediction of all the predictions that were submitted in the project, the competition. So it was a really interesting case study. And as the authors examined all of the results that came in, how the compounds performed compared to the predictions or intuitions of the medicinal chemists who had worked on these projects for years, they sort of broke down several different categories or classes of results. In one case, we've got the sort of at the bottom left of our chart here, the class 0, endpoints that are neither correlated nor important. We expect a lot of these, right? Not everything will be related to everything else. We might say nothing to see here. On the other side of the top right of our plot, we have what we might call class 1 relationships, where significant relationship between these two endpoints is identified by both methods. The endpoints are both correlated and important. And we might call this the I could have told you that, right? An experienced chemist could have looked at this and said, yeah, I expect that to be a correlated relationship. Terrific. In class 2, we see toward the bottom right. Here we see highly correlated endpoints that don't rise to that threshold of importance in the deep learning imputation model. So this is a case where we might say, well, that's odd. There's a bunch of things that seem correlated, but only one or two of them is predictively important. And we'll unpack that more in the coming slides. And then finally, at the top left of our graph, we have what we might call class 3 relationships. In this case, there really is no underlying correlation, nothing that would be detected by our other methods. And in fact, things that are often missed by the chemical intuition that the model highlights and says, no, this relationship is really important, very predictive for your endpoint of interest. C and I'll call these the machine overlords, right? In the case of that medicines for malaria study, the compound that Optibrium submitted was in a chemistry space that had been explored by the medicinal chemist. They'd tried most of the functional groups. They never thought that the compound submitted could be active. And yet it turned out to be the most active compound submitted in the project. Right. So sometimes these methods can capture relationships and help us identify things that we would miss from our other knowledge of the situation or from standard correlation analyses. Okay, so let's take a look of our top 10 most accurately imputed endpoints. For two of them, we see lots of class 1 relationships across the data. We see the points clustered here at the top right of our graph, meaning that these endpoints are both important and correlated. The correlation analysis and the deep learning imputation are identifying and using the same relationships, the same endpoints. We see also a lot of class 2 relationships. So here, 5 of those top 10 most accurately imputed endpoints show significant class 2 relationships in understanding what's important and predictive. So here we see, right, the reason these are in class 2, we have lots of points to the bottom right below the diagonal. So this means there are correlated endpoints, but not all of them rise to the level of imputed importance, right? Significance for that imputation model. We can highlight a few of these relationships here. And we see that in many cases, there's a cluster of points, a cluster of endpoints that all show similar correlation, but only one or two of those rises to the level of predictive importance for the imputation model. So what is it that's going on here? Well, the points below the diagonal are generally of comparable size or smaller. The size of the point here is giving us an indication of the amount of experimental data that was available for that endpoint in the data set. So smaller points will have less experimental data available, which will naturally result in typically less predictive or imputative power. So 82% of these sort of class 2 endpoints, the ones that aren't used by the imputation model, contain less data than the most important or most predictive endpoints. So this is one of our key themes here throughout the presentation today. Matt's mentioned this before. We'll see it here and we'll draw it together again at the end. One of the powerful features of deep learning imputation with Cerella is that you can use fewer experiments to get the same predictive value. Then finally, we want to look at a few of our endpoints that show some class 3 relationships. This is where things get really interesting, and it's almost astonishing to see the things that the deep learning imputation can pick up. So again, these class 3 relationships are looking at nonlinear relationships, right? So endpoints that are not correlated or maybe identifiable by other standard analyses. But Cerella in the deep learning imputation has picked up on these relationships and says, actually, this is really important. So let's break these down one at a time. At the top left, we're considering what endpoints are important for predicting the properties of mouse liver microsome half-life data. And the endpoint that's most important for that prediction is the human liver microsome clearance. So what we see is that Cerella's deep learning imputation detects and leverages interspecies relationships, which would be hard to identify maybe in other approaches, but is able to leverage that for its predictions. Now, of course, if we think back to the way that Matt laid this out at the beginning, this isn't the relationship that is of most interest to us. It's very rare that we would want to use human experiments to impute mouse outcomes, right? It's much more likely we would want to do the other. And that's what we see in the graph on the top right. So now we're looking at what endpoints are predictive for human liver microsome clearance. And we see mouse liver microsome half-life is important, very important for predicting that relationship. So again, the Cerella's deep learning imputation is detecting and leveraging those interspecies relationships. We've labeled the point at the bottom right of this graph. This is the mouse liver microsome clearance. And we see this is highly correlated, right? We expect strong correlation between the mouse liver microsome half-life and the mouse liver microsome clearance. But that strong correlation within species isn't adding extra information to the predictive power of the imputation model. And so even though there's that strong correlated relationship there, Cerella's deep learning imputation is picking up only the most important, the most predictive, and leaving aside other highly correlated data. Finally, we'll look at an example on the bottom left. This chart on the bottom left is what relationships are important for predicting log D. And here we see a very uncorrelated but important the fraction unbound in mouse plasma protein binding. Again, as we just saw above, this is not the direction in which we want to impute this relationship, right? We'll never want to go do mouse experiments to predict a property like log D. What would be really interesting is if this went the other direction. And so we did. We went and found the predictions for the fraction unbound in mouse plasma protein binding. And sure enough, this relationship goes both directions. And log D, though very, very weakly correlated, right? Essentially uncorrelated data, is very important for imputing that fraction unbound in the mouse. What's going on here? Cerella is likely picking up indirect correlations among ADME endpoints, right? So there's not a direct correlation here between the two endpoints, but we know that, as Matt said at the beginning, those in vivo or PK kinds of endpoints, the plasma protein binding here, will arise from a combination of other factors and earlier physical chemical properties. And we see here Cerella is able to leverage that relationship and pull out for us the importance of the log D for being able to accurately predict the fraction unbound in mouse. Okay, so that concludes our little case study here on the Medicines for Malaria Venture pathogen box data. I'd like to take us through just one other example, a case study that we published with Genentech, where we were looking at a much bigger set of data. So in this case, we were looking at using Cerella's deep learning imputation to profile a kinase program, where we've got hundreds of kinases and thousands, more than 11,000 compounds. So a huge set of data, again, as Matt drew our attention to at the beginning, very sparse data, right? So if we think of 11,000 compounds and something like 600 kinases, the number of experiments that we've actually made for that data matrix is quite small. And so we'd like to say, well, can Cerella help us to identify which are the most meaningful relationships, the most important measurements that we can make to prioritize our experimental resources? This would let us save time and money by running only the most important experiments. And to give away the end in the beginning, what we find out is that Cerella's deep learning imputation was able to achieve higher performance in predicting test set data than random selection, using data from 76% fewer assays. So we'll unpack this just a little bit. The graph here at the top right is showing we've built a model over the kinase data, and we've held out a test set so that we can evaluate the performance of the model. We can see that if we use the whole test set for imputation, we get the same results regardless of the method. So the experiment here was to sort of pull back and say, well, how little of the test set can we use to impute the remaining test set values and still get a reasonable model? And so you can see as we start to use only subsets of the available data, we have a choice. We can let Cerella identify the measurements that will be most important for enhancing the predictive power of the model, or we can randomly assign data to the different sets and let Cerella model the data that we've given it. Here you can see that if we let Cerella choose the most meaningful data, the models are more accurate. And you say, well, maybe that's only moderately more accurate. And that's true. It's an interesting and helpful result, but the really surprising and astonishing and powerful result is if we look at what data is being selected. At the bottom right here, as you might expect, if we give a random selection of data, we're plotting a histogram. So how many points of experimental data are used for any particular endpoint? And so here we can see almost all endpoints have a little bit of data drawn from them in the random approach, which is what we would expect. The left bar graph, if we look at what Cerella has chosen for its model building, we find out that 76% of the assays have no data selected for that model. So all of the data is coming from just a very small subset of the available endpoints. And so here's what that looks like if we consider that overall matrix, the overall matrix of possible experiments, possible relationships. We can see on the left the typical pattern of measurements for these kinase assays. When we filter out using only the optimal selection of data by Cerella, we find that there really are just a handful of endpoints that we can measure to give confident predictions of the rest of the data set. So again, this is really important and ties back to what Matt started us off with on the examples that we've seen in our little case study. And here we demonstrate again, it is possible through Cerella's deep learning imputation that we can save time and money by focusing only on the most important experiments. So just to conclude what we've seen through the case study here today, first, we hope that we've shown that deep learning imputation using Cerella provides more value than correlation analysis alone. We saw those 4 different classes of results where really the class 2 and class 3 results, we see Cerella adding value that we couldn't get from correlation. Cerella, with the deep learning imputation, can leverage our existing data to maximize the accuracy of predictions. Cerella can help us to use more, less expensive early stage data to confidently impute the more costly late stage measurements. And it can help us to avoid missed opportunities. So again, we've demonstrated the explainability of these results with Cerella. We've been able to show cases, right, what we call class 1, where the results are easily identified by both methods, correlation or deep learning imputation. But in class 2, we find that Cerella is using only the most predictive endpoints. This is that real savings that we were talking about in the kinase program, identifying only the ones that are really essential to measure in order to give good predictions of the rest of the data. And then, of course, in class 3, we saw that Cerella can pick up indirect, nonlinear correlations, relationships that we might miss. We would certainly miss from correlation analysis, and we might even miss from experienced intuition of chemists who have worked on the project. Cerella is able to detect those relationships and present them for us to help us understand better what's happening in our system. So with that, I'm going to conclude and thank my colleagues at Optibrium who have helped with this case study, and our partners at Intelligence, as Matt said, with whom we collaborate on the Alchemite model under our Cerella platform.
Yeah, say thank you, Daniel, for a really fascinating study and sharing those results. I just wanted to say a few words before we move on to the Q&A session. I just talk a couple words about Cerella. We've mentioned it a few times during today's presentation. So what is it? It's a proven and deployable AI platform that empowers drug discovery teams to do a number of things. To go from concept to candidate faster, as we described, using early stage data to confidently predict late stage expensive outcomes, moving those compounds faster through the process and finding high quality compounds for your candidates. It does help to identify better compounds, finding those hidden opportunities caused by missing, uncertain, or incorrect data, and helping to comprehensively explore optimization strategies for synthesis and testing. And as we've been focusing on today, it also helps to make informed decisions, choosing the most valuable experiments to guide compound progression, and also helping to understand the underlying mechanisms of action and adverse outcome pathways for your compounds um, and your objectives. It also helps to automate that process of building and validating models. So, it's time consuming, expensive to constantly be updating and building these models as new data become available. So Cerella automatically updates and prepares experimental data. It connects directly with your data source, your data mart, your ELN, your database. It populates that sparse matrix that we've been discussing today. And then it uses the Alchemite method to build models covering all of those different experimental endpoints. And of course, it will then fill in those blanks using the imputation method that we've been talking about. And it will provide seamless access to all of those results, all the models, all of the interpretation we've been talking about via our Stardrop platform, but also via other platforms if you wish. It has a RESTful API, so if there's other ways you want to analyze or visualize those results, you're very welcome to. This is about making sure you can access these really sophisticated methods very, very easily in a very user-friendly and easily supportable way. And make sure that you're always working with results based on the latest information, the latest data that you're generating. We've alluded to a number of examples and case studies. We've had the privilege of working with all different sizes and shapes of organization, primarily on the sort of the drug discovery and pharma side of things, but also in non-pharma applications, for example, flavors and fragrances with IFF and agrochemicals with FMC. And we'll give you a link, a QR code where you can find webinars and publications describing all of these different applications across the discovery process. And with that, it is now over to you, Catherine, to talk us through the Q&A and to manage those questions we've received. So thank you very much.
Sorry for being so keen to get to all of your questions. So a reminder, just hit the Q&A button if you have any more questions that come to mind. But we'll get started. So the first question here, Matt, maybe you can tell us something about how the importance values are calculated.
Yeah, absolutely. So just dig in a little bit deeper into what Daniel described. So the first thing that it looks at, of course, is the strength of the relationship between the different endpoints. And as Daniel nicely demonstrated, that doesn't have to be a linear correlation. It can also be non-linear relationships as well. But there are other factors one needs to take into account as well. One, again, Daniel mentioned this, is the amount of data available for each endpoint. The model has to understand what data it can expect to have. There may be a very strong correlation between two endpoints, but if there's only a handful of data points for one, it can't reliably be used to impute another. And the third thing is the uncertainty in the data, both the variability in the experimental data used as input, but also where the greatest uncertainty of prediction lies. So we want to be able to leverage those inputs that have the most information with which to impute those most uncertain outcomes that we're trying to predict.
Great, thanks, Matt. Next question, I think, Daniel, you might be able to take. If you have multiple correlated inputs, how does Cerella choose the most important?
Thanks, Catherine. Yeah, this really is, in many ways, the the same three things that Matt just drew our attention to. Right? We showed that in 82% of the cases, it was explainable just as which endpoints had the most data available to use for building that imputation model. But in some cases, right, as Matt described, we need to consider the overlap of the data, right? Is the data meaningfully overlapped so that a relationship can be derived that will be predictive and useful? And then finally, there are some cases where it will really be the uncertainty that will tip the scales over what's most predictive. If you have a lot of data, if you're not confident in that data, it would be less useful in terms of predictive power. So it's really drawing those three things together in each individual case that Cerella identifies which relationships give the most predictive power.
That's great. Thank you, Daniel. Our next question, Matt, you might be able to answer. Would a non-monotonic association measure, such as mutual information, perform better than Pearson's linear correlation?
That's a really great question. So the reason that Daniel looked at Pearson correlation coefficient, if I may speak for you, Daniel, is that's the first question that's always asked as well. Is it just picking out the obvious linear correlations? And so that is a really good sort of baseline and the most common analysis that people apply. But you're absolutely right that you can look for nonlinear correlations using a variety of methods, including mutual information, and it will pick out, I guess, more of the relationships because it will find those where there is a nonlinear correlation that wouldn't be identified by a simple Pearson's correlation coefficient. However, there's more that you can get out of this imputation process as well. So it is any correlation method typically is a sort of a one-to-one. You're looking at the correlation between just two endpoints. But actually using this sort of nonlinearity and this imputation method, you can identify situations, for example, where endpoint A and endpoint B are not individually strongly correlated with endpoint C, but the combination of A plus B or some linear combination or even nonlinear combination of A and B would correlate with C. And so it can find these indirect correlations, I think that Daniel also spoke about as well. And so, yes, there's much more information than one can gather where we can leverage that in making more accurate predictions and also understanding those relationships.
That's super. Thank you, Matt. Next question. For the kinase example, the targets that were selected from the imputation model, were these the best performing models or was it a mix?
Maybe I'll take that one as well because I don't think Daniel did a lot of work directly on that project. The answer is it was a mix. So it's obviously trying to find the specific kinase activities that have the most information content with which to impute the full matrix of the full profile of activities. Some of those will come from where we've got a great model. But actually, a lot of them will come from where you don't have a good model. If we had a great model, we would just use that to, impute the other missing data. It's actually where you have a poorer model, perhaps, that experimental data adds the greatest information. So it's a little bit of both.